Welcome back everyone. In part 1, we understood some basic questions. What is genetics? What is heredity? What is variation? What is DNA? And why is DNA called the blueprint of life? But now a very interesting question comes. How did scientists discover all this? How did humans discover that characteristics are inherited? How did scientists discover DNA? And how did they finally understand the famous double-helix structure of DNA? Today, we are going to travel through the history of genetics. Imagine living hundreds of years ago. People could see that children resemble their parents. They could see that plants produced similar plants. They could see that some diseases appeared repeatedly in families. But they didn't know why. There was no concept of genes. There was no knowledge of DNA. There was no molecular biology. Scientists had observations. But they didn't know the mechanism behind inheritance. Now we come to one of the most important names in genetics. Gregor Mendel. Mendel was an Austrian monk and scientist. He performed experiments on pea plants. And his experiments became the foundation of modern genetics. That's why Mendel is commonly called the father of genetics. Now you might ask, why pea plants? Why didn't Mendel experiment on humans? Obviously, experimenting on humans wasn't practical. Pea plants had several advantages. They had clearly visible contrasting characteristics. For example, tall and dwarf plants, yellow and green seeds, round and wrinkled seeds, purple and white flowers. They also have a relatively short generation time and can be self-pollinated or cross-pollinated. Mendel crossed plants having different characteristics and carefully observed their offspring. For example, tall plant times dwarf plant. He didn't simply look at one generation. He studied multiple generations. And he counted the offspring. This was extremely important because Mendel used mathematical analysis to understand inheritance. Mendel proposed that hereditary information is passed through discrete units. Today we understand these units as genes. He also described important principles of inheritance, including law of dominance, law of segregation, and law of independent assortment. We will study these laws in detail later in this masterclass. Here's another interesting part of the story. Mendel published his work in the 1860s. But his work was not immediately appreciated by the scientific community. Later, around 1900, scientists independently rediscovered the importance of Mendel's work. This helped establish Mendelian genetics as a major field of biology. Now scientists knew that inheritance existed. But one major question remained. What physical material carries hereditary information? Scientists knew that cells contained different molecules. But which molecule actually carried genetic information? Protein? DNA? Something else? This became one of the biggest questions in biology. Now let's talk about an important experiment. In 1928, a British scientist named Frederick Griffith conducted experiments using bacteria that caused pneumonia in mice. He worked with two forms of the bacterium: S strain, smooth colonies and virulent; R strain, rough colonies and non-virulent. The important observation came when heat-killed S bacteria were mixed with live R bacteria. The R bacteria were transformed into a virulent form. Griffith called this phenomenon transformation. Something from the dead S bacteria had somehow changed the living R bacteria. But Griffith did not identify the exact chemical responsible. He only showed that some transferable, transforming principle existed. Now scientists wanted to know, what exactly was this transforming principle? In 1944, Oswald Avery, MacLeod and McCarty provided strong evidence that the transforming substance was DNA. Their work showed that when DNA was destroyed, transformation did not occur. This strongly supported DNA as the genetic material. Another famous experiment came in 1952. Scientists Alfred Hershey and Martha Chase worked with bacteriophages. Bacteriophages are viruses that infect bacteria. They wanted to answer a simple question. When a virus infects a bacterial cell, does the virus inject DNA? Or does it inject protein? They used radioactive labels. DNA contains phosphorus. Proteins can contain sulfur. So they used radioactive phosphorus DNA and radioactive sulfur protein. They allowed bacteriophages to infect bacteria. Then they separated the viral coats from the bacterial cells. The radioactive phosphorus associated with DNA was found inside the bacterial cells. The radioactive sulfur associated with protein remained mainly with the viral coats. This provided strong evidence that DNA, not protein, is the genetic material in those viruses. Now scientists knew DNA was the genetic material. But another question remained. What does DNA actually look like? What is its structure? How can one molecule store so much information? Scientists began studying the chemical composition and physical structure of DNA. An important contribution came from Erwin Chargaff. He studied the amounts of different nitrogen bases in DNA. He found an important relationship. The amount of adenine is approximately equal to thymine, and the amount of guanine is approximately equal to cytosine. Now we come to a very important scientist, Rosalind Franklin. She used a technique called X-ray diffraction to study DNA. Her work produced important evidence about the structure of DNA. One of her famous X-ray diffraction images became known as Photo 51. Using experimental evidence from multiple scientists, James Watson and Francis Crick proposed a model for the structure of DNA in 1953. They proposed that DNA has a double helical structure. Imagine a ladder. The two long sides of the ladder are the sugar phosphate backbones. The steps are formed by pairs of nitrogen bases. Now twist the ladder. You get a structure resembling a spiral staircase. That's the basic idea of the DNA double helix. In 1962, James Watson, Francis Crick and Maurice Wilkins were awarded the Nobel Prize in Physiology or Medicine for discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material. Rosalind Franklin had died in 1958 and therefore could not receive the Nobel Prize. Now let's create a timeline. This is extremely useful for exams. 1860s. Mendel P. experiments. 1928. Griffith transformation. 1944. Avery, MacLeod and McCarty DNA evidence. 1952. Hershey and Chase DNA as genetic material. 1953. Watson and Crick double-helix model. One important thing. Don't think that one scientist discovered everything alone. Science is a cumulative process. Mendel helped us understand inheritance. Griffith showed transformation. Avery, MacLeod and McCarty identified DNA as the transforming principle. Hershey and Chase provided strong evidence for DNA as genetic material. Chargaff provided base relationships. Franklin's X-ray diffraction work provided structural evidence. Watson and Crick proposed the double-helix model using available evidence. So today we traveled through more than 100 years of genetics research. We started with Mendel and his pea plants. Then we saw Griffith's transformation experiment. We learned how Avery and his colleagues identified DNA as the transforming principle. We saw how Hershey and Chase provided strong evidence for DNA as genetic material. We learned about Chargaff's base relationships. And finally, we reached the famous double-helix model of DNA. But we still haven't answered the most important question. What exactly is DNA made of? What are its building blocks? What are A, T, G and C? Why does A pair with T? Why does G pair with C? And how can this tiny molecule store an enormous amount of biological information? That's what we are going to discover next. In part 3, we will break open the DNA molecule and study its structure from the smallest building block, the nucleotide, to the complete double helix. Thank you, everyone. Keep learning. Keep questioning. And remember, science becomes easy when you understand the concept. This is Concept Science.